Today I'm going to talk about ecological drivers of carnivoran body shape evolution. So one of the most distinctive patterns across the tree of life is the diversity of body shapes. And several researchers have quantified the major axes of body shape variation in primarily squamates, fishes, and now mammals. And these studies often find that body elongation is the primary axis of body shape evolution. Variation in body shape facilitates morphological and functional innovations that can lead to niche specialization. For example, extreme body elongation allows Australian skinks to burrow in sand faster compared to more robust bodied species. Body elongation in moray eels allows them to exhibit innovative nodding behavior during prey capture. And body elongation in ferrets enables them to hunt prey in crevices and burrows while lowering their metabolic costs. Biologists have also begun to examine relationships between body shape variation and ecological diversity at the macroevolutionary scale. Researchers have found relationships between body shape variation and ecological diversity, for example, in lungless salamanders and in teleost fishes. However, these empirical studies have produced conflicting patterns about the adaptive evolution of different body shapes across different clade levels. For example, there is no relationship between body shape variation and habitat use when looking across all salamanders. And recent work found that body shape is more diverse within each habitat for teleost fishes as opposed to between habitat types. In mammals, relationships between body shape evolution and ecological diversity is virtually unknown. Mammals exhibit a diverse range of body shapes, but what generates this diversity has not been tested. Therefore, the goal of this study is to examine if selection drives mammals with similar ecologies towards similar body shapes. In this study, I used the mammalian order Carnivora because of its high species richness, morphological diversity, and ecological diversity, including locomotion that ranges from arboro to fully aquatic, hunting behaviors, and trophic levels, everything from hypercarnivory to herbivory. To examine these relationships between body shape and ecology, I quantified body shapes using head-body elongation ratio, or ER. And I calculated ER by adding the head length and the total body length of each individual and dividing it by the rib length as a proxy for body depth. And I used measurements based on osteological specimens held at natural history museums. So I did this for 685 specimens across 207 species, which is about 74% of the total diversity and includes all 16 carnivoran families. I then categorized each of those species into one of 6 locomotory modes, one of 6 hunting behaviors, and one of 5 dietary regimes. So here's a simple violin plot of ER of the 16 carnivoran families. So we can qualitatively say that there's quite a bit of body shape variation both between and within these different families. Now I want to see how ecology influences this body shape diversity. Specifically, does locomotion, hunting behavior, and diet influence the body shape allometry? And second, do those ecologies then influence or drive body shape evolution? So looking at body shape allometry first, here we have a simple plot of body size on the x-axis and ER on the y-axis. And using a PGLS, I found that body shape scales with negative allometry with respect to size. This means that larger species exhibit more robust bodies, whereas smaller species exhibit more elongate bodies. The mechanical demands of terrestrial locomotion probably contributes the most to this allometric trend. Larger animals require more robust bodies to support their heavier weights, whereas smaller bodies might be free to evolve elongate forms that can facilitate flexibility for prey capture in crevices and burrows and other small spaces. Now I'm going to show you the same plot with the 3 different ecological groups overlaying that. And using phylogenetic ANCOVAs, we can quickly see how the aquatic to terrestrial axis influences body shape allometry. Species with aquatic locomotion, hunting behaviors, and diets exhibited positive slopes, whereas species with terrestrial ecologies exhibited negative slopes. And a possible explanation for this positive trend in aquatic species is the balance between drag reduction and thermoregulation they must manage within the aquatic environment. Streamlined bodies reduce drag, but also leads to heat loss. However, relatively larger fossils can overcome these disadvantages because of their relatively lower surface area to volume ratio, and therefore may have the luxury of exhibiting a more elongate body that is more closer to the optimal fineness ratio without compensating those thermoregulatory needs or drag reduction. So now moving on to see if these ecologies then drive body shape evolution. So to test this, I fit evolutionary models. And these models allows us to examine if selection from different ecological regimes led to different body shape optima, or if it was just random variation. So the first model I fit was a Brownian motion model, which models a random walk process in which the variance of ER increases linearly through time. The second was an Unstein-Uhlenbeck model, which constrains ER to evolve towards a single optimum through time. And these two models suggest that there is no selection towards different body shapes between ecological regimes. The other three models I fit were multi-peak OU models that test if selection drove ecological regimes to evolve distinct ER optima. So I fit three multi-peak OU models that test for distinct ER optima between locomotion, hunting behavior, and diet regimes. The last model I fit was a multi-peak OU model that does not include a priori groupings of ecological regimes because additional factors or phylogenetic natural history may influence body shape evolution. So I compared the fit of these 6 models using AICC weights, and I conducted simulations to determine whether I had enough power to distinguish between these models. So using this selection approach, I found that the multi-peak OU model without a priori ecological regimes was the best fitting model. And this model reveals that there are 4 independent shifts towards more elongate bodies and 3 independent shifts towards more robust bodies. And for those who are curious, the second best model was the multi-peak OU model with hunting behavior regimes, suggesting that maybe hunting behaviors also influences the evolution of body shape. However, parametric bootstrapping reveals that these optima were largely overlapping between the different regimes, indicating that the optima between hunting behaviors are not really distinct. Together, these results indicates that carnivorans with shared locomotion, hunting behavior, or dietary ecologies do not evolve towards similar body shapes. And there are 3 possible explanations for this decoupling between body shape variation and ecological diversity that I came up with. The first is trait multidimensionality, second is allometry, and the third is phylogeny. So first, the multidimensionality of body shape may conceal the one-to-one mapping of morphology onto ecology across these broad macroevolutionary scales. And several types of form to function relationships have been hypothesized to drive this or explain this decoupling. The first is many-to-one mapping of form onto ecology, which suggests that multiple body shapes are functionally equivalent, enabling species with distinct body shapes to exploit similar resources and also exhibit similar ecologies. For example, American badgers, black-footed ferrets, and swift foxes are sympatric carnivorans with distinct body shapes that feed on prairie dogs in the same grassland habitats. Equally possible is the one-to-many mapping of form onto ecology, where a single functionally versatile body shape can exploit diverse resources and not be restricted to a specialized resource. For example, the American mink exhibits an elongate body plan, but is able to hunt in a variety of habitats, including chasing rodents into burrows, birds on the surface, or actively swim after fish in the aquatic environment. Lastly, many-to-many mapping suggests the multidimensionality of both morphology and ecology creates complex relationships between body shape and ecological diversity, in which one-to-one, many-to-one, and one-to-many mappings can all occur simultaneously within a clade. So essentially, anything can happen. Alternatively, allometric patterns driven by body size may be more important than specific ecological traits. I found that other than the aquatic to terrestrial axis, all other ecological regime exhibits identical allometric patterns. Specifically, the mechanical demands of gravity drives negative allometry in species with terrestrial ecologies, whereas the balance between drag reduction and thermal regulation probably drives positive allometry in species with aquatic ecologies. Lastly, the best supported hypothesis I had that may explain this decoupling between body shape and ecology is that the evolution of carnivoran body shapes is primarily driven by phylogenetic history rather than ecological adaptations. And this is consistent with previous findings that carnivoran families served as evolutionary significant units and that the disparate evolutionary processes often drive differences in morphological disparity between carnivoran families. For instance, when you think of a bear, they all exhibit that similar bear-like body shape, despite their diverse ecologies. And that low diversity in body shapes may be conserved through evolutionary history. In contrast, mustelids exhibit evolutionary shifts towards more elongate body plans along their clade that contributed to their diversification. So clearly, there are different processes between different clades of carnivores. Nevertheless, the next steps are really to incorporate the fossil record to better understand the evolutionary processes that drove the evolution of distinct body shapes, both between and within Carnivoran clades. And with that, I would like to thank Charlene Santana, John Flynn, and Rob Voss, as well as my funding sources and all the museum collections for letting me use their specimens for this research. You can read more about this research in these two publications in Evolution and American Naturalist.